So I'm just going to give you 3 examples then about how we're using XRF in the biomaterials field. In this first example, this is a paper where we created a 3D model of bone to replace the traditional 2D tissue cultures. And what we essentially do is that we have a Petri dish filled with a gel called fibrin, which can be manufactured from 2 blood components. We then place 2 calcium phosphate anchors in the middle, and we can lace these gels with osteoprogenitor cells. So these are the precursor cells for bone formation from humans. And these are highly contractile cells. So when you put those cells in the gel, they like to wrap the gel around them, kind of like a blanket. And because of the lines of tension there, the gel contracts and contracts and contracts until you form this band of material, which is about 3 millimeters thick and about 10 to 12 millimeters wide. And over that time then, because you're providing that 3D matrix that's roughly similar to what happens in the body, those precursor cells start to differentiate, and they lay down bone, and they start to construct the same or similar 3D microenvironment of bone that you'd normally see in the body. And what we'd expect to see is for that whole structure to be replaced by bone mineral over time. And indeed, that's what you can see here. Between 7 days and 3 months, you can see the structure start to densify over time. So these are just micro CT scans, just showing in bulk what we'd like to see. And what we want to be able to see is the fibrin matrix dying away over time, being replaced by this bone mineral in red. But from a chemical perspective, just knowing where the calcium phosphate isn't enough on its own. We want to understand how is that bone mineral maturing over time. And this is where we use 2 techniques. So before we used to just use Raman on its own, which is a technique for looking at, we use it for looking at organic species, but we look for how atoms are bonded together to make different phases of calcium phosphate. But we use XRF here as a rapid screening tool. So we want to look inside these hundreds of different bone models that we make and understand where is the mineral and how much of it is there. And indeed, what we can see here is that if it's 100% mineral, we'd expect the red and the green parts of the map to completely co-localize to form yellow. And we don't quite see that. We often see areas of rich calcium and rich phosphate. So that on its own is an area of interest to us. We want to understand why are there different forms of calcium phosphate in there. What we use from the Raman is once we can identify where these calcium hotspots are, we can use Raman to probe them in greater detail. And indeed, once we know where the calcium phosphate is, we can find out what phase it is. And this is really important to us because these 3 phases here, octocalcium phosphate, hydroxyapatite, these are early and later markers of bone maturation over time. And here we can visually see how the bone is maturing from this dark red to purple color to the more greeny mature bone over time. From the XRF map, we can quantify level of bone mineral in a non-destructive manner, and we can monitor this over time. But the key for us then, why XRF? If we can do this, if we could look at bone mineral and look at different phases from the start from Raman, why are we even bothering to use XRF in the first place then? The key for us is that although Raman is non-destructive, technically, because we're looking at such low concentrations of mineral embedded in a really thick, highly fluorescent matrix full of proteins, it's really difficult to get a good signal-to-noise ratio. As a consequence of that, we had to scan our samples often for 24 to 30 hours at a time, and then it became destructive. You just destroy the sample over time. And that's not really scalable. We can't really be making hundreds of these in one batch. We need to be smarter about how we characterize the samples. What XRF allows us to do is instead of spending 24 to 30 hours scanning a whole sample looking for mineral, we could scan the same sample for for around 30 minutes to look for where the mineral hotspots were, and then just zoom in with the Raman on the areas of interest, and therefore still preserve the sample. The other reason why we wanted to switch to spectroscopic methods of looking at bone minerals, in our field traditionally staining has been king. So here is alizarin red, which chelates the calcium, and that's used as a surrogate marker for where mineral is. But just compare so red here is where there's believed to be mineral. But look here, there's hardly any staining at all after 7 days, which would suggest there's no mineral at all. But in reality, in XRF, we can see very early mineral deposits. So in other words, the staining is not really reliable and is actually quite subjective based on the lighting conditions that we use. And that's pretty critical for us, because if we just relied on that data, we wouldn't have understood how the bone forms at a very early stage. So I think in summary there, XRF gives us a really quick, early window into quite subtle chemical changes into tissues as they mature over time, and it's quite part of our workflow. But now we've validated XRF as a method for looking at quantifying bone mineral density, we can turn it on its head now. And now we have a model for bone formation, we can now screen potential drugs that could prevent bone formation for drug companies. and we can quantify how they inhibit bone formation over time in the space of a day. So these are a tray of samples, so we can scan the whole tray in one go overnight, rather than one by one. So actually you've got your own internal control there. In the second example then, so this is where we're creating a cell therapy. And this is really to exploit the fact that we can actually have a farm of these osteoblast cells, which could kick out early matrix vesicles. And what we want to do is, can we farm thousands, trays of these cells and harvest these early mineral bone matrix vesicles and inject those as a therapy in patients who struggle to have fractures healed over time. So in this experiment then, we had cultures of osteoblast cells, and we want to analyze what are the cells kicking out. How many vesicles are they kicking out? What is in those vesicles? How advanced is the mineral in there? So we would collect the media and we would spin it down to form a pellet and dry it. And at the top here, so actually just want to focus on these two. So BMP2 is a current gold standard growth factor for stimulating bone. It's used in spinal fusion. And this is one of our early prototype therapy, vesicle therapies for stimulating bone. And what we did see in the end, what we were hoping to see actually, is lots of yellow in terms of lots of calcium and phosphorus co-localizing. What was really odd, however, is that we don't hardly get any co-localization at all. We almost get independent calcium and independent phosphorus, and we totally didn't expect that at all. Because under the current model, we were expecting early mineral formation, so it should co-localize. So XRF actually opened up a bit of a Pandora's box for us in terms of bone formation, and kickstarted another 3 years of work. So why didn't the calcium and the phosphate co-localize? Well, actually XRF then gave us 3 regions of interest to look at. This calcium-rich region, this yellow region, and the phosphate-rich in the background. Actually, when we dug into these regions of interest using FTIR, which could tell us again the phase of this mineral and how that mineral associates to proteins in the system, so clear on here. But roughly what this set of figures shows is that there is a really early mineral form called octocalcium phosphate, but actually that doesn't associate with the protein background hardly at all really in this region. What we do know is that most of that raw calcium is actually bonded to a phospholipid, and that's quite a key finding for us because what that told us is that in those vesicles there isn't calcium and phosphorus bonded together to form mineral. There's another protein that's keeping the calcium and the phosphorus apart inside the vesicle. That was key for us because that explains why your cells and your tissues don't mineralize on their own automatically. So it's kind of like a safety mechanism. And we wouldn't have found that had we not been able to differentiate localization of elements and compounds overall in these samples. As a final example then, moving more into the material side, here are 3 examples of how we're engineering effectively sugars, gellan, to deliver molecules around the body. So in the top diagram here is a picture of our dressing, which contains an anti-scarring molecule called decorin. Here is a schematic of how we've engineered an eye drop that can set on the eye to keep a drug on the surface of the eye, again to prevent scarring. And this is an injectable formulation to keep a stem cell population in a cartilage defect. 2 of these are going to trial. Actually, WMG could probably appreciate this, but if we're going to get anything into the clinic, we have to have really robust quality control methods. Now, we don't manufacture the clinical grade at university. We build the manufacturing process and we port that across to a clinical facility. But also, we need to understand what's coming off this manufacturing line. Is it within tolerance? And actually, how does the chemistry of the material change during manufacturing, which has been the source of a lot of big hiccups in the past, actually, when it comes to adverse reactions in the clinic. But we have a really big challenge with some of our products. Particularly with some of our scar prevention products, the actual drug itself, which we don't engineer. A lot of the drugs themselves are biologic based, they're really expensive to make, and in fact one that we're helping to reformulate costs about £200 for every mil. Now, if we have a quality control test that requires 10 mil, that's a lot of money wasted. Not wasted, that's a lot of money, it's a lot of product taken off the production line. And we can't really afford to do that, and neither can the pharmaceutical industry. So we have to be really smart about what techniques we use to analyze changes in chemistry in the material overall. They're quite pragmatic actually. You can use pretty much any technique you want in the eyes of a regulator to look at changes in chemistry as long as you can validate it. The problem for us though, from a biological perspective, there's lots of assays out there which indirectly measure concentration or indirectly measure the form of something, but the reliability is often called into question. In other words, whether they can cross-react with other analytes in the system. So Where we've used XRF then really is, in fact this is quite a good example. So this is a prototype, this is our first batch of eye drop that we took off the production line. And normally if we were to look at contaminants that cause precipitation, you can see them by eye. So you can see the system go cloudy. This particular batch looked cloudy upon visual inspection, but we did a random test on one of our products. And we actually found then that we had a lot of early stage precipitates distributed throughout the gel itself, and these were instigated by manganese. Now we would never have spotted that in our old quality control system. We did using XRF, using maybe about 50 microliters of material. What's the significance of using mapping to do this rather than any other technique? Well, if we look back at the way we make our eye drop, for example, our eye drop is made essentially by making a colloid of polymer and then subjecting that polymer to quite high shear such that your polymer chains, which are floating in solution, crumple up into gel particulates. So all the cross-linking agents are enveloped within those particulates. Now, by seeing the manganese localize in these little patches here tells us that the contaminant must have come from the cross-linking agent and not the water we were using. We would expect the manganese to be freely distributed in the background there. So just by looking at how the contaminants are distributed allowed us to pinpoint where exactly in the manufacturing process we were likely to have an issue going forward. So in summary then, we see XRF as complementary to things like RAM and FTIR. It fits within quite a large workflow for looking at tissue chemistry and biomaterials chemistry over time. And we use XRF really as a scoping tool really to map either a large quantity of samples or a large area in one go. And then using things like RAM or FTIR or even sometimes TM as well as a sniper really to focus in on areas of regions of interest that often take quite a long time to map. The key advantage for us, particularly in our mineralization work, is that the big downfall of a lot of our older techniques that relied on sample fixation and dehydration. Well, you can think that if the cell has got lots of free calcium and phosphorus, what's the downside of dehydrating there? You can end up getting condensation of those phosphorus, which for us could risk giving us a false positive for mineralization. So we wanted a system where we could look at the tissues of the samples in as fresh as native condition as possible. We wrote a paper on our findings from some of the drawbacks we found in the area and how they can all work together over time. But going forward, again, XRF doesn't replace any technique. If anything, it allows us to work smarter. So we still use synchrotron XRF, we still do synchrotron diffraction studies, but XRF has allowed us to narrow down from 100 samples down to 3 really good ones and use our synchrotron time a lot more smartly overall.